Your body performs millions of chemical reactions every second. Often, reactions this frequent require high temperatures to sustain their speed, yet your internal environment is locked at 37 degrees Celsius. Biological catalysts, known as enzymes, allow these processes to occur at lower temperatures. These specialized proteins catch a specific reactant, or substrate, and speed up the reaction without being consumed themselves. Without these catalysts, essential processes like digestion and cellular respiration would move too slowly to sustain life. We can map this process on a graph of energy versus reaction progress. Every chemical reaction requires an initial energy investment. Before reactants can transform into products, they must overcome a specific energy barrier. This barrier is represented by the height of the hill. We measure this from the starting baseline to the peak and call it the activation energy. This threshold acts like a hill that every reactant must climb before the reaction can proceed. Without an enzyme, activation energy is 50 kilojoules per mole. Few reactants clear this high hill. Enzymes provide an alternative pathway, lowering the barrier to 20 kilojoules. Enzymes don't add energy. Starting and final energies of reactants and products remain exactly the same. The enzyme only changes the height of the energy barrier. By making the hill easier to climb, the reaction can proceed much faster at 37 degrees. Enzymes are highly specific. For instance, amylase breaks down starch. The protein folds, creating a uniquely shaped active site. They lock together, forming an enzyme-substrate complex. The starch breaks into products, and amylase releases them unchanged, ready for the next reaction. This specific three-dimensional structure is the only reason the enzyme can function. If the protein loses its shape, it can no longer bind to its substrate. Reaction rate climbs as heat increases, causing more frequent collisions. It peaks at the optimum temperature, 37 degrees. Past this, activity drops sharply. Excessive heat breaks bonds, physically warping the active site. This is denaturation. The enzyme can no longer hold the substrate. This makes heat a double-edged sword. It increases the speed of collisions until it reaches a point where it destroys the protein's precise structure. Now, consider how pH affects reaction rate. Pause and predict the graph in highly acidic or basic environments. Unlike temperature, the pH graph forms a symmetrical bell curve around its optimum pH. Extreme acidity or alkalinity disrupts protein bonds, denaturing the active site. Because chemical extremes on both ends of the scale result in denaturation, the graph shows a balanced decline on both sides of the optimum. These three models summarize how enzymes function. The energy profile shows how they lower the activation energy, while the temperature and pH graphs show how environmental changes impact the reaction rate. When describing these effects in an exam, always link the drop in reaction rate to the physical denaturation of the enzyme's active site. Understanding these relationships explains how external conditions like temperature and pH determine whether the chemical reactions necessary for life can occur.